Okay, let's start. So in this video, we will see the types of PCR, different types of PCR. Okay, so let's start our video. So first type is reverse transcriptase or RTpcr. cDNA is synthesized and it is used to study gene expression. So let's see this in a whiteboard to properly understand. Suppose there is a RNA. Okay, there is a RNA. From this RNA, we will reverse transcribe this RNA into cDNA by using reverse transcriptase. By using reverse transcriptase, we will convert this RNA to cDNA. Okay, then this cDNA will be amplified by PCR. Then cDNA will be amplified by PCR. This is known as RTpcr. That means first what we did, we reverse transcribe our RNA and then we perform PCR. That's why it is known as RTpcr. First we reverse transcribe, then we performed PCR. Okay, reverse transcribe and PCR. So here cDNA is synthesized. Okay, this RTpcr is used to study gene expression. It is used to study gene expression. So let's see how RTpcr is used to study gene expression. We know that a gene expresses itself by producing RNA. Okay, gene expresses itself by producing RNA. Suppose there is a gene A. Okay, gene A expresses itself, so it will produce RNA. Okay, then we will convert this RNA to cDNA by using reverse transcriptase. Okay, this cDNA can be amplified by PCR. So product will amplified. Okay, PCR product will be amplified. This amplified product can be visually seen. This amplified product can be visually seen by gel electrophoresis. By gel electrophoresis. This amplified product can be visually seen by gel electrophoresis. In the second step, suppose gene A do not express itself, so it will not produce RNA. Okay, if there is no RNA, it will do not synthesize cDNA. It will not synthesize cDNA. If there is no cDNA, then there will be no amplified product. There will be no amplified product, and it can be visually seen that there is no amplified product. So if there is amplified product, that means gene has expressed itself, and if there is no amplified product, that means gene has not expressed itself. In this way, by using RTpcr, we can analyze gene expression. Okay, next is real-time PCR. Next is real-time PCR. It is also known as quantitative PCR based on fluorescence emission. Trackman probe is one probe. Markers are used, allows to analyze PCR in real time. So let's understand this also in whiteboard. Generally what happens, other types of the product of other types of PCR can be analyzed after PCR is completed. But in real-time PCR, we can analyze during the process is occurring. Means when the PCR is running, at that real time, we can analyze it, whether PCR is occurring or not, whether our required segment is present or not, whether it is amplified or not. We can analyze during the reaction. Okay. So in real-time PCR, we use some probes. That are fluorescently labeled. that are fluorescently labeled. We use probes that are fluorescently labeled. Okay. So what happens? Suppose these two are DNA strand. Here forward primer and reverse primer is anil. Okay. In probe, okay, in probes, two molecules are attached. One molecule is known as quencher, and the other molecule is known as reporter. When quencher and reporter both attached to the probes, the reporter will not emit emission. The reporter will not emit fluorescence. It will not show fluorescence. When reporter and quencher attached to the probe, the reporter will not emit fluorescence. Okay. So when primer binds to the specific sequence, then probe will bind in between them. Okay. Probe will bind in between forward and reverse primer along with quencher and reporter molecule. Okay. If this segment is amplified, by tag polymerase or DNA polymerase, then probes get degraded. Okay. When probes get degraded, the quencher and reporter get separated, and when the reporter gets separated, it will emit fluorescence that can be seen. Okay. Probe is attached to reporter and quencher, means a quencher molecule and a reporter molecule will be attached to probe. Okay. So it will bind between the forward and reverse primer. Okay. Probe will bind to forward and reverse primer along with reporter and quencher. When DNA extensions take place, then DNA polymerase will degrade the probe. As a result, quencher and reporter will be separated, and reporter will emit fluorescence. And this can be seen. Okay. That means when extension is occurring, then we can see the fluorescence at real time. That's why it is known as real-time PCR. Okay. There are different markers are used in real-time PCR apart from this Tachman probe. Okay. This is Tachman probe. Like cyber green is there, molecular beacon is there. So these are different types of marker. In this example, what we have seen is the example of Tachman probe. Tachman probe. Okay. Reporter and quencher. Okay. When it is degraded by DNA polymerase, then reporter and quencher get separated, and reporter will emit fluorescence. Okay. The next PCR is multiplex PCR. Amplifies several DNA sequences simultaneously, uses multiple primer. So let's understand this in whiteboard also. Multiplex PCR. So in multiplex PCR, suppose different types in PCR tubes. Okay, this is a sample. Okay, we have taken the sample in PCR tubes, and if we want to see whether segment A, gene segment A, gene segment B, and gene segment C is present in this sample or not, then what we will do, we will take this sample and we will synthesize primer for gene A, segment A, primer for gene segment or gene B, and we will also synthesize primer for gene segment C. Then we will add this primer in these PCR tubes. Okay, we will add this primer in PCR tubes. If gene segment A is present, then we will get an amplified portion of gene A or gene A segment. If B segment is present, then we will also get an amplified portion of B segment. And if C segment is also present, then you will get an amplified portion of C segment. Okay, that means multiplex PCR will give us a massive data, massive output, and sometimes it becomes difficult to analyze the result because it will show the presence of different segments. Like here A segment is present, B segment is present, and C segment is present. So sometimes it becomes difficult to analyze the result. It is multiplex PCR. Okay. Next is nested PCR. Nested PCR reduce unwanted binding of primer, uses two sets of primer, It is important, two sets of primer into successive run. Second run amplifies target within the first run product. Second run amplifies target within the first run product. Okay, let's see this here. So this is a double stranded DNA and this is our target sequence. Okay, so cycle 1, DNA melting and primer binding. Here first set of primer has been binded. First set of primer is binded here and here. Okay, so this primer will amplify this portion, and from this primer the amplification will take place up to this. Okay, this primer will amplify up to this, and this primer will amplify up to this. See here, amplification has been done up to here and amplification done has been here. Okay, here amplification is done up to this and amplification up to this. We do not need this, so it is cut it out. Okay, see, first set, the amplified product of the first set of primer include our target sequence. Okay, first amplified portion of this first set of primer include our target sequence. Then these two strands are again separated. And we add the second set of primer. Where we add second set of primer within the amplified product of the first primer. Within the amplified product of the first primer, we have added the second set of primer. Now this primer will amplify only our target sequence. It will not synthesize unwanted or untargeted sequence. It will not synthesize. The second set of primer that attached within the product of the first set of primer will amplify only the target sequence. That means nested PCR will increase the specificity. Okay. Then colony PCR. Bacterial colonies are screened directly by PCR. Colonies are sampled with sterile pipette and small quantity of cell are transferred into PCR mixer. DNA is released by extended denaturation step. So let's understand colony PCR whiteboard. Okay. Colony PCR. Okay. Suppose this is a nutrient agar plate and here different colonies have grown like this. Okay. Different bacterial colonies have grown in this nutrient agar plate. Okay. In conventional PCR, first what we need to do, we need to extract the DNA. Then this extracted DNA is PCR amplified. Okay, this extracted DNA is PCR amplified. But in colony PCR, what we do, by using a sterile pipette, we directly pick this colony and transfer into a PCR tube. we directly pick these colonies and transfer into a PCR tubes. Okay, we transfer this into a PCR tubes. Then after transferring this, the colonies are there. Okay, now if this was a conventional PCR, then we need to extract the DNA from this bacterial cells. But in colony PCR, we do not need to extract the DNA. What we do, we just increase the timing of denaturation step. We increase the timing of the denaturation step. By increasing the timing of the denaturation step, the DNA from these bacterial isolates come out. Okay, DNA of this bacteria come out, and then PCR runs. Okay, then PCR runs. This is known as colony PCR. Okay, we directly transfer the colony to PCR tube, then we increase the denaturation step to release the DNA, and then DNA is PCR amplified. Okay, the next is hot start PCR. Here non-specific amplification is lowered by heating the DNA melting by increasing the temperature, by increasing the melting temperature. Here non-specific amplification is lowered by increasing the melting temperature. Okay, then touchdown PCR. Here initial temperature is 3 to 5 degree higher than melting temperature and is gradually decreased. Okay, it provides greater specificity. Okay, let's see this in a while. Generally annealing temperature is 3 to 5 degree lower than melting temperature. Generally annealing temperature is 3 to 5 degree than melting temperature, but lower temperature increases unspecific binding. Lower temperature increases unspecific binding. That's why in touchdown PCR, what we do, we increase the temperature 3 to 4 degree. We increase the temperature 3 to 4 degree above melting temperature, so that specificity increases. And after that, we gradually decrease the annealing temperature in subsequent cycling. Annealing temperature is then gradually decreases. In first annealing, the annealing temperature is 3 to 4 degree higher than melting temperature, and in the next step the annealing temperature is lowered. Okay, so this is colony PCR. This is this is touchdown PCR. Okay, so these are different types of PCR. This is nested PCR. So if you find our video useful, then please like the video, share among your friends. Okay, and also subscribe the channel. Okay, thank you.